that would allow for selective activation or placement of bacteria to produce complex images. So we want to be able to control the expression of the bacteria through the use of chemical inducers or control the localization of the bacteria. So we first decided to focus on controlling the expression. Our construct was going to be a plasmid which would have three, okay, here we go. Oh, let me go on. We would have a plasmid that would have three inducible fluorescent proteins controlled by separate chemical inducers. And then we would distribute the chemical inducer in a controlled manner to produce a complex image. So one potential endpoint that we focused on was using an inkjet printer to distribute the inducer. So basically we would inject the inducer into the inkjet printer cartridge, and then we would transfer the inducer via some transfer mechanism, such as placing it onto a plate, or excuse me, onto paper, and then this would then be transferred onto a plate with a bacterial lawn. The benefits of this that we felt were that you could get incredibly fine precision. Of course, there's issues with diffusion and other various, outside things that are a little hard to control, but that was one of our primary focuses. And then in terms of the genetic end of that, as I said, we'd have three independent operons to control the expression of the three different fluorescent proteins, and then they would be applied to achieve maximum resolution. So this is just a brief overview over mechanism of induction. So just go through that. So basically, here's our three primary parts that we started with. We had the red fluorescent protein M cherry controlled by the Lux operator. So this is a sort of an inducible fluorescence using this operator because there is some small constitutive expression through the Lux promoter, but it can be increased using homoserine lactone or some other similar derivative. The second one was using the TET operator with the cyan fluorescent protein. And then the third one was the LAC operator with the yellow fluorescent protein. And the TET would be induced by anhydrotetracycline, and the LAC operator by IPTG. We actually got, most of our primary parts came from the registry, so that was a huge help in getting started. Here's an overview of what I just said. And of course, there's some issues with the parts being sort of leaky, as we'd say, but that's just something we had to deal with. Here's sort of a list of the composite parts we made. We have included the fluorescent proteins with the conjugated promoters, and then also we produced the conjugated promoters, constitutive expressed repressors, and inducer complexes. And then we have our composite watercolor design, which would be the complex repressors and inducible fluorescent proteins. So I talked briefly about the printer, but we explored a few more options in terms of distributing our inducers to the cells. One's probably the most simplistic method would be just pure pipette application, which we used a great deal in quantitating our bacteria. The third would be to simply grow them in the inducer to get similar to having a constitutive expression because they are completely in contact with inducer. The third was the inkjet printer, which I overviewed. And the fourth was a PDMS stamp, basically rapid prototype a negative, and then we produce a PDMS stamp, which we could then use to, as the name implies, stamp the inducer onto the bacterial lawn, producing an image. Okay. And then Brian will give the second part. Thanks.
Okay, so once we had our final construct done, we set out to characterize the best way to induce the proteins and how to look at them. We used ultraviolet light imaging and also fluorescent microscopy. And so we attempted to induce them in two different ways to find the best concentrations and application methods. The first way was just to make a lawn of bacteria, and we just had a little array on the agar plate. and then varied concentrations of inducer put onto the lawn from the km to 10 to the 6 km. And then from there, pipetted a drop into each spot on the array, and then imaged them at various time periods after that. And the second way we induced them was to grow up the cells in a LB broth with the inducer in there at various concentrations, and then pipette them onto the plate from there after mixing, and grow them up and look at them the next day. So here's some of the images we got with fluorescent microscopy. What we found was these are taken with a 10x objective, so you're looking at whole colonies there. And we found that some colonies would have very strong fluorescence, or even these patterns, which I wasn't sure if that was from motility of bacteria leaking out the fluorescent proteins, or just fluorescent proteins maybe leaking out and moving around. But some colonies would fluoresce very strongly, and some very weakly or not at all. Here's another picture of that. So as you can see, these are all the same genotype, but very different phenotypes. We weren't sure what that was coming from, whether it was instability in the plasmid or whatnot. And we also found that regardless of the concentration of inducer applied to the cells or whether there was inducer at all, we got the same results. The same amount of, or relatively the same amount of colonies fluorescing on a given plate, and with the rest not fluorescing. And this is an image of, the difference between these two images is this is a lower seeding density, so you can see between the colonies, whereas this was a very concentrated bacterial solution when we plated it, so it was one large colony. And here's another image of that. We even saw with some colonies, you have these divisions within the colony itself where we would lose the fluorescent protein expression. So our results with our final construct were that there was not a quantifiable or observable difference between the induced and non-induced bacteria. So they were just doing what they wanted to do. And we had single colonies exhibiting very strong fluorescence in yellow, weak in M cherry, and we never really saw anything that we could pick up off the background with the cyan fluorescent protein. Although it should be noted the cyan is a very weakly fluorescing protein in itself. I think it's about 30% of what the original green fluorescent protein was, whereas the yellow is much stronger. We used the enhanced yellow actually. So that could be why we weren't picking up the signal, or the cells actually weren't producing the protein. So at this point, we decided to go back to our ideas of methods to place the bacteria onto the lawn, and we concentrated on using the inkjet printer, and we used our intermediate constructs as our final construct was not working correctly. So our initial idea was to replace the ink in the printer with the chemical inducers, and then print the inducers and use those to stamp a lawn of bacteria and express the proteins. At this point, since we weren't getting expression with the inducers, we decided to see if we could print the cells onto a piece of paper and then use that to stamp onto the agar medium. So we just used a regular HP OfficeJet printer. It was nice because it has separate cartridges for each color. Took the cartridges out, just basically removed the ink. There's a septum right here in the bottom of the cartridge. Removed the ink with the syringe, rinsed them multiple times with just DI water until we couldn't get any absorbance in the spectrophotometer in that color. Go ahead. So then at that point we replaced the ink with a solution of cells and water, not in the agar, fearing that we'd get clogging from the sugars in the agar. So we actually just scraped cells off a plate, added them to water, vortexed it to mix it up, injected in the cartridge, and this is actually a picture of the print head. So you have these lines running from the print head to the inkjet cartridges, which it takes quite a bit of time to get from the inkjet cartridges, which are in a different spot in the printer, to the actual print head. So we found there's septums there as well that we could use to prime the whole system with the cells that we wanted to print. So then, as I was saying, we printed the cells onto a transfer paper, and we just used regular cardstock paper. And just used because you can't see the cells on the paper, we just put basically a black circle around the image so we knew what to cut out and how to place it onto the auger. And then stamp the auger and place them in an incubator. So here's some of the images we got. This is a baseball player. And this is our university logo. So you can see there's some overspray, which could actually be, I mean, it could be when we were stamping the auger, it's a little bit inefficient because you can move it around and it'll smear. So we lost resolution there. Or it could have been overspray from the print head because of the liquid properties of the water versus the ink. And we have some more images as well. So and there's our university logo. And there it is again. So.
Okay, so in terms of, this was actually our first iGEM project, so we learned a lot. Starting out, we had basically less than no idea going in. But for future iGEM projects, we learned that we need to start planning much sooner than we did because we started right at the beginning of summer. But it was a great experience in terms of leading a team. We definitely learned a lot about how to do that, and we'll have a lot of lessons for future projects. And for us, the focus was the integration of the biology and the engineering, more so than just focusing on the biology, as you can see with our printer application. So we were extremely excited about that. And so for future iGEM projects, we've learned a lot, and I think next year we're going to come back with a much stronger team. So we also learned about the viability for the printer model for fine cell distribution. We're still working with this, and it's really exciting, but of course we need to do some refinements to where we can get it to do maybe an experimental work, maybe converting the printer to be able to print directly onto the auger or other things. But as you can see, we got images, so we were excited. Okay, so the unpredictability, this sort of goes to our first idea. Our first final construct was the inducible bacteria. And so as you saw from the pictures of the rather weird-looking colonies, biology is not, a perfect science. It's not math where A plus B equals C. So in terms of that, we weren't sure if it was plasmid instability or what, but having a single genotype of cell and then getting out diverse results, that also informed our ideas for future iGEM projects. So in the future, if we're not getting exactly what we want, we might know it's not necessarily our fault. Or maybe it is, and we just... All right, and right here's our acknowledgments. We had an incredible amount of support from our university and the associated personnel. Most of them aren't here, but and we also had incredible support from our faculty advisors. They did a great job in helping us run the project and lead the team. So that's our...
So we've got about 10 minutes for questions. If anybody has questions?
Yes.
How did you choose your printer, your printer model? Is that a thermal inkjet system?
No.
Yeah, I believe it's a thermal inkjet system, and we chose it initially because it had the separate colors and separate ink cartridges. And that was really the only...
Do you think, and the cells survived
Yeah, cells
the
survive.
heat of the thermal explosion through the orifice?
Enough of them did.
You might want to consider a piezo.
Yeah, we were in
inkjet
the future.
going forward, which one heated them up to that high a temperature.
Yeah, back there.
As far as that goes, the one that's actually boiling the liquid is boiling a small amount of liquid which pushes out a slug of
Bursts of bubble.
So it's not hurting those cells. And then you also need to look at new change printers. Some of the new ones have really tiny nozzles on there. So if the cell gets crosswise or something like that, you might start having more clogging than the older printer with the large size nozzles.
Yeah, we actually did have some issues with clogging, quite a bit of rinsing and trying different solutions and solvents and whatnot. So that was actually the biggest issue was with the printer was your first run through, you get really good, nice images, and the next run through, you get a few cells. So, but we are still working with that on ways to keep from clogging. I think the best thing for that would be to print directly onto Augur, because we could lower our concentrations of cells that were actually running through the print heads very low, whereas printing onto the paper and then transferring to the Augur, we had to use fairly high turbid concentrations.
Just got curious about the medium you printed onto. You're printing onto regular office paper? Or
Cardstock paper, yeah, from Kinko's.
Printed down the autoclave with stacks of paper? How would you prepare the paper for being printed onto?
Well, we printed onto the paper and then cut out the image and stamped it onto a petri dish.
We didn't actually autoclave it, so that's a possibility.
I was just curious. Like, you said,
before printing.
On the autoclaves with stacks, just going, what?
Yeah, we actually didn't have any issues with contamination, most likely because it was the LV amp Augur.
Yeah.
Just a quick point about your fractal images. Those are most likely due to mutations in plasmids. We've seen that with plasmids that tend to mutate rather quickly. I mean, we can actually, for example, correlate that with FACS measurements. For unstable plasmids, they would tend to have these kind of fractals. You'll notice that it actually represents the growth of cells.
Right.
Actually, what happens is the cells are not producing as much fluorescent protein, will tend to grow a little bit faster, and that's why you see the regions kind of enlarge over time. One thing that ends up being useful, and then goes back to your notion of biology not being predictable, is that what you want to do is take the colonies that are supposed to be identical to everything else, and the ones that are highly fluorescent, you'll take those, and those will actually tend to maintain the plasmid more stably over time.
Actually, we did do a screening process where we had these images and we had fully fluorescent colonies. We plate them, and then we'd get the various phenotypes. And I mean, it could just been the initial plasmid was unstable, and maybe those colonies had. But
yeah. So you can play around with things like reducing the copy number of the plasmid and so on.
Yeah, we did quite a bit of streaking and selecting cells, and I believe in the end we did get a strain, if you will, that had a higher rate of fluorescence, but there was still no difference between the induced and non-induced cells.
And that made
which induction system?
For all of them, for all the different fluorescent proteins. In other words, we would streak the cells and select the strongly fluorescent colonies, and the colonies fluorescing strongly with inducer, as well as the colonies not fluorescing without inducer.
Using replica plating or something?
Just streaking, so you get isolated colonies, and then if you put them in the UV box, you can see which ones are fluorescing and which ones aren't. So we tried taking that and in parallel taking the colonies that would not fluoresce without inducer, and then working with those to see if we can induce them to fluoresce, and then taking the ones that were strongly fluorescing with inducer and trying to get them to not fluoresce without inducer, but we didn't get the results we were looking for.
So we had issues with our
Would the cells grow if you just lay the paper down face up with the paper just on top of wet Augur?
Yeah, we tried that. The issues were that the paper, either there was too much media in there and all the media would become inoculated and the cells would spread over everything, or there's not enough media in there and the paper everything would dry up.
Yeah, we were considering trying, putting the paper on top of a sponge and trying some diffusion, but maybe something to do in the future.
But I would think, yeah, that would be a distinct possibility, and probably you would get the better resolution because you don't have to, like I say, transfer it. I mean, you're just doing it with our hand, kind of transfer and try not to move it around too much and then pull it off. So I'm sure there's some smearing there is why those images weren't as sharp as they could be.
Up here in front.
Which part of the ones that you put in the BioBricks registry, which one do you think was most critical to your work and you're most proud of?
Well, I guess, I'll talk about two parts. One will be our final construct, which we got those kind of cool fractal images, and just because that's what we were working towards, but it didn't work. So I guess the second favorite would just be our constitutively expressed cherry protein, because it worked and we got pictures from it. So that'd be it.
Any other questions? Okay, let's thank our speakers.